ഷിജു ടീച്ചറെ, ഷിജു ടീച്ചർ.
സാറേ പറഞ്ഞോളൂ, കേൾക്കാം.
അല്ല ഷിജു ടീച്ചർ, നമുക്ക് തുടങ്ങായിരുന്നു.
സ്റ്റാർട്ട് ചെയ്യാൻ പോവാ. ഓക്കെ ആണ്.
ടീച്ചറെ, ഷിജു ടീച്ചർ.
വൈഷ്ണവിയാണ്. വൈഷ്ണവി ജോയിൻ ചെയ്തിട്ടുണ്ടോ?
ഷിജു ടീച്ചർ, നമുക്ക് സ്റ്റാർട്ട് ചെയ്യാം. എന്നാൽ
സ്റ്റാർട്ട് ചെയ്യാം സാറേ. വൈഷ്ണവി Vaishnavi.
Good morning. Good morning to one and all present here. Welcome to the 8th day of ILLUMINISMO 2021 webinar series on emerging trends in interdisciplinary sciences and social sciences. Today, the Department of Life Science is organizing the webinar on the topic Advanced Technologies for Vision Restoration with the eminent resource person, Dr.Biju B Thomas. He is Assistant Professor in University of Southern California, US. Welcome you all to this webinar. The presence of all is essential for a bright start. Hence, I would like to invite Ms. Nandana Vinod for a short prayer.
ഭൂമി തളിരിടുന്ന നമ്മിൽ വാഴുമാതിനാമം ഇന്നു വാഴ്ത്തിടാം. കൃപയരാഗതന്ത്രി നീക്കി സ്നേഹഗീതമേറ്റിടും ഭൂമി തളിരിടുന്ന വർണ്ണമേറ്റിയും നമ്മിൽ വാഴുവാതിനാമം ഇന്നു വാഴ്ത്തിടാം. ഞങ്ങൾ പാടുമീ സ്വരങ്ങൾ കീർത്തനങ്ങളാക്കണേ, ചൂടുവെക്കുമീ പദങ്ങൾ നൃത്തലോലമാക്കണേ, കുഞ്ഞുവേദികൾ സ്വർഗമാക്കണേ. അമ്മ നൽകുമമ്മ പോലും അമൃതമാക്കണേ. പൂർണ്ണമീ ചരാചരങ്ങൾ ഗുരുവരങ്ങളാക്കണേ. പൂർണ്ണമീ ചരാചരങ്ങൾ ഗുരുവരങ്ങളാക്കണേ. ഹൃദയരാഗതന്ത്രിമേറ്റി സ്നേഹഗീതമേറ്റിയും കർമ്മഭൂമി തളിരിടുന്ന വർണ്ണമേറ്റിയും നമ്മിൽ വാഴുവാതിനാമം ഇന്നു വാഴ്ത്തിടാം. നമ്മിൽ വാഴുവാതിനാമം ഇന്നു വാഴ്ത്തിടാം. Thank you.
Thank you, Ms. Nandana Vinod. Now, I would like to invite Dr. Shiju Jacob, Head of the Department of Life Science and Computational Biology, to welcome the gathering.
Respected Principal Sri Biju Joseph Sir, our resource person Dr. Biju B Thomas, and all the dignitaries and delegates who have gathered here in this online platform. A very good morning to one and all. With immense joy and pleasure, I am here to welcome Dr. Biju B Thomas, Assistant Professor of Research Ophthalmology, University of Southern California, US. Before I venture officially to the duty of welcoming all of you, let me say a few words about our department. St. Vincent College is established in Rajapuram, Kasaragod in 1995, and in 2020, after 25 years, as a new generation course, BSc Life Sciences and Computational Biology started as the 7th UG course with 24 students. The core course consists of 2 subjects, zoology and computational biology, with microbiology and computer science as complementary elective subjects. Today on 10th August, our department is organizing a webinar as a part of ILLUMINISMO 2021, an international webinar series on emerging trends in interdisciplinary sciences and social sciences from 3rd to 13th August 2021. First of all, I express my sincere gratitude to Dr. Biju, because when I asked him about his willingness to share his views without any hesitation, he accepted our invitation. Now it is around 9 p.m. there in California, and after a day's work, he is spending his valuable time with us. He is one of my good friends. Moreover, we are family friends. I wholeheartedly welcome Dr. Biju B Thomas to share his words with us on advanced technologies in vision restoration. Next, I welcome our principal, Sri Biju Joseph sir, who always support us in every programs we conducted in our college. I also welcome Dr. Sinur Kariyach, convener of the webinar series, leading us in a dynamic way, and also welcome the technical team of for their entire work. Next, I welcome Ms. Jinsimol Joseph, Assistant Professor in Computer Science, who will be introducing our resource person, and Dr. Cellular Gopalakrishnan, Head of the Department of Microbiology, who will be proposing the vote of thanks in this session. Next, I welcome all the dignitaries and delegates who have joined with us this morning to attend the webinar. Thank you, one and all. Thank you.
Thank you, Shiju ma'am, for those warm words of welcome. Now I request Mrs. Jinsimol Joseph, Assistant Professor of the Department of Computer Science, to introduce the resource person, Dr. Biju B Thomas, and welcome him to lead the webinar.
A very warm good morning to all. Today, our resource person is Dr. Biju B Thomas, discussing about advanced technologies for vision restoration. Dr. Biju B Thomas, Assistant Professor, Research Ophthalmology in University of Southern California, US. His clinical specialties are Retinitis Pigmentosa and age-related muscular degeneration. Our research topics of Dr. Biju are brainstem electrophysiology, visual behavioral testing, and stem cell-based therapies. And about his education, he got PhD from University of Kerala in 1997 and postdoctoral fellowship from University of Louisville in 2001. And also, Dr. Biju have many publications based on his research area. I'm sure Dr. Biju is an art person for today's session. So I welcome Dr. Biju B Thomas to handle this session. Thank you.
Okay, thank you very much.
Thank you, Jinsi ma'am. Now, I would like to invite Dr. Sinos Karyachan, Assistant Professor, Department of Microbiology, for moderating the session.
Moderation will be after the talk only. Now, Biju, you can.
Okay, all right, thank you. So let me share my screen with you. Just want to make sure you can see it clearly.
Yes, sir. Okay.
1 second. Okay, 1 second. Okay, good morning, everyone. Today I'm going to talk about advanced technologies for vision restoration. So just to give you more information about myself, I work in the University of Southern California. I run a research lab that consists of postdoctoral fellows and other research staff. And my lab is mostly focused on therapies for retinal degeneration diseases, basically for curing blindness. And I was lucky to get involved in 2 different projects that already went to clinical trial. So one is based on stem cells and the other one is based on retinal prosthesis. So when I go into this deep into the talk, many of the research results what I'm going to show you probably would be not very easy to understand, maybe because it is too much for many of the undergraduate students and even for master students. So I will try to make it as simple as possible. So anyway, before starting the talk, I would like to first give an introduction to the structure of the eye. You can see now on the screen a human eye, and the whole structure of the eye is shown on the left side. That shows the cornea, pupil, the lens, and inside you can see that a reddish, yellowish, large area, which is the fluid-filled area, that is the vitreous. And on the back of the eye, you can see the blue color retina, which is the most sensitive part of the eye, which is more like it's a part of the central nervous system. Even though eye is separated from the brain, it is highly similar to the various structures we find in the brain. Especially the retina contains the neurons, different layers of neurons and photoreceptors. So basically the light enters into the eye through the pupil, goes to the back of the eye, it falls on the retina, and on the right side you see an enlarged structure of the retina. It's very important to know this structure, otherwise it's very hard to understand the rest of my talk. So on the right side, you can see that in the enlarged picture, there is an arrow showing how the light is entering, falling on the retina, and inside the retina, you can see on the extreme end of the right side, you can see the red and green and blue colored cone-shaped structures. Those are the photoreceptors. Basically, the light passes through all these structures. If you look at the left side again, coming back to the whole structure of the eye, you can see that the pupil, the hole through which it's just like a camera shutter, the pupil. The light enters through the pupil, fall on the back side of the eye, falling on the retina. Then look at the right side, the enlarged picture. The light enters into the retina. It passes through the different layers of the retina, reaches the back of the extreme interior of the retina, where you see that photoreceptors arranged in layers. These include the rod photoreceptors and cone photoreceptors. And just the last layer, you can see a dark layer there. That layer is called the retinal pigment epithelium. And outside the retinal pigment epithelium, there is circulatory vessels are there. So basically, the retinal pigment epithelium receive nourishments from the blood circulation, passes it into the inner retina, provides support for the photoreceptors. The photoreceptors, at the same time, it's a highly metabolite area in the body. So it produces a lot of waste materials that are taken by the retinal pigment epithelium and removed from the body through the circulation. So this structure needs to be well understood before we move further, because everything what I'm going to talk about later today is about the structure of the eye. Because all the diseases that affect the eye, either it affect the neurons here, affect the photoreceptors here, or affect the retinal pigment epithelium. So when the retinal pigment epithelium, for example, if the retinal pigment epithelium is dysfunctional, it cannot support the photoreceptors. At the same time, if it is dysfunctional, it cannot remove the waste materials. So just because of this layer is damaged, the photoreceptors can be degenerated or diseased. When the photoreceptors are degenerated, the connections with the inner neurons will be also damaged. That will lead to the damage to the various neurons inside the retina. So in a normal condition, the light falls into the photoreceptors, that the photoreceptors will produce the neural signals into the neurons. All the different neurons have their own properties. I'm not going to go to the details of those properties, but that's how we see the various things. And then all these neurons terminate into the innermost neuronal layer called retinal ganglion cells. Those retinal ganglion cells forms the optic nerve, and optic nerve will take the information to the brain. So if an optic nerve damage happens, the person can become blind. If the retinal pigment epithelium is damaged, that can eventually lead to photoreceptor loss and blindness. If the photoreceptors are dysfunctional, that will also lead to blindness. So there are a variety of different disease conditions or accidents that can lead to the blindness. So blindness is a very condition which cannot be easily reversible. So, and it largely affects the quality of a person. So curing blindness is one of the top priorities, especially in the developed countries. On the top of that, there are also age-related diseases that also causes blindness. So above the age of 50 to 60, there are diseases called age-related macular degeneration that causes blindness in a large number of people worldwide. In addition to that, there are other disease conditions where congenital diseases where due to some genetic defect, the photoreceptors can be damaged in an early stage. That will also lead to blindness. That kind of diseases are called generally called retinitis pigmentosa. Is there any question or anything?
Dr. Thomas, we will be going the question answer session at the end of the talk.
Okay. So after this introduction, I will go into the details of the various treatment strategies, what we are working on. So again, I want to inform you that my laboratory is mostly focused on treating various disease conditions based on different approaches. Stem cell is a major topic of my research, where we try to replace the retinal pigment epithelium or to replace the photoreceptors. Those are the two approaches we use. There are other approaches that include retinal prosthesis, where we don't need any photoreceptors. The artificial retina or a chip is placed inside the retina that will be directly connected to a camera for getting the images out from outside and transmit to the brain. I will give the details later. Then there is another approach in which we also try to make any kind of cells in the retina light sensitive when the photoreceptors are damaged. So there are different approaches. So let us go through those approaches again. But before that, I just want to show you diseases which are very common among people. One is retinitis pigmentosa and second one is age-related macular degeneration. So when you look at this picture, there is you can see difference across everything. So this is what is called a fundus image. When you go to eye clinic, they have special microscope to take the picture of a. So just before this slide, I showed the pupil. A camera that goes through the pupil can take the image of the inside the eye. So this is what that pink colored area I showed you before in the previous slide. So, but other than that, it mostly shows the back of the eye. It's not showing anything interior, but just showing the blood vessel. And this is optic nerve head, and this is the central vision area called the fovea. So you can, when you look at these three pictures, you can see that there is a difference because there are pigments accumulating. There are dots here. All this shows diseased condition. So this is called, this particular condition is called retinitis pigmentosa. In the bottom, just look at the two cute boys holding soccer balls, and how they, for a person with retinitis pigmentosa, how this they will see the kids. They could see only the face, but not the peripheral area. So this is called a tunnel vision. That's a hallmark of retinitis pigmentosa disease. On the other hand, age-related macular degeneration, they cannot see the central area, but they can see only the peripheral area. So the face is not visible, but the peripheral area is visible. So they are losing the acute vision. So there is a reason for that, because that depends on if the highly sensitive area, the fovea is damaged, then the central vision is lost. But if the peripheral area is damaged, then the peripheral vision is lost. So the mechanism of the disease is different. The retinitis pigmentosa is, most of the retinitis pigmentosa diseases are genetic defects where photoreceptors are unable to produce the light triggered signals and send the neural signals because of their phototransduction defects. Whereas in age-related macular degeneration, the disease is mostly related with the aging process during which the retinal pigment epithelium is no more able to support the photoreceptors. So either this disease condition, it is important that we need to develop treatment strategies to cure these diseases. So when we, again, coming back to the different treatment options. So the first one, which I underlined and given in red font, is replace or restore lost photoreceptors. So that is mostly by using cell therapies. You might be familiar about stem cell research, which is getting very important nowadays. So stem cell is a major hope for the first option, that is replace or restore the lost retinal photoreceptors. The second option is bypassing damaged retinal layers. So that is mostly by using artificial electronic chip placing inside the retina. There are different types of these kind of prosthesis chips. They can be either placed on the surface of the retina, it is called epiretinal, or it is in the inside the retina, it is called subretinal, or on the other side of the retina, it is called a suprachoroid. So different type of retinal prostheses are developed and even being used for human clinical trials. So the first option is stem cell or cell replacement therapy. Second one is using retinal prosthesis. And the third one is making the remaining non-light responsive retinal cells light sensitive. So there is a way you can do it. For example, if the photoreceptors are completely lost, there are other cells inside the retina, and there are techniques like including gene therapy and using viral vectors, you can introduce factors into the eye that will, so these factors would be light sensitive, just like the photoreceptors. And in that way, you can restore the lost vision. So there are three totally different approaches. So that's what I'm going to discuss more. In fact, I was lucky to be involved in all three different approaches. And as I mentioned before, the prosthesis is already in clinical trial that I was one of the contributors for animal studies. And also one of our stem cell product is already in the clinical trial, where again, I was mostly involved in the preclinical studies using animal models. And I was also involved in a non-light responsive retinal photosensitive, making the remaining non-photoreceptors light sensitive project. But unfortunately, we were not able to move it to a clinical trial phase for various reasons. Anyway, so let us, so again, I want to reinstate that my laboratory, so the research in US is unlike back in India, it's highly collaborative. So each and every project, not a single person or a single laboratory can handle everything. So all our research projects, especially those goes into the clinical phase, trial phase, there would be involvement from different laboratories that involve biomedical engineers, stem cell experts, and experts in animal studies, clinicians, surgeons. So a lot of different hands are contributing to major research projects. That's only way a successful project can go to like a clinical trial phase. So so each and every laboratory, they might have a focal area, but still they might collaborate with the different people to support their projects. That's why I'm saying that my laboratory is mostly focused on stem cell therapy, where I lead my people to work on stem cells. I submit research projects mostly related with the stem cells. I obtain external funding for conducting research. And the other situation is some other laboratories, for example, retinal prosthesis, they have engineering section and they apply for grants, they get money. But when it comes for animal studies, they might come to me. So I help them. So those kind of collaborative projects are mostly moving into the clinical trials. Again, starting with the stem cells, how the stem cells, transplantation of stem cells can work. There are two different ways it can work. One is the trophic factor effect, where when you introduce healthy cells into the retina, these stem cells especially, they produce a lot of neurotrophic factors. We say like probiotic factors. You can call it in any way, but it's mostly the neurotrophic factors that is released by the cells. They can help the remaining cells in the retina to rejuvenate. And the good thing with this kind of an approach is this is not something intentional, but sometimes it end up that any kind of stem cell therapy also contribute its effect through trophic factors. But the good thing is there is no question of any neuronal integration. So once the cells are delivered inside the retina, they have the capability to, as long as they survive, they continue to release factors that help the remaining cells in the retina to thrive, and also they will be rejuvenated and they will be more active. Then the second is a direct cell replacement effect, which is more difficult to accomplish because of the requirement for neuronal integration. So I'm just showing some tools which are used for, you can see that these are all schematic diagrammatic representation of how a human eye and rodent eye, their cell transplantation is conducted using these kind of tools with a sharp nozzle that goes into the subretinal space where the cells are released. So this is just showing some histology images of how when after you transplant cells, how the retina looks like. You can see that as part of the retina looks like it is normal shape. And also on the right side, there are again fundus, that is what the image of the eye inside, showing how the cells are, the retina is recovering after several months after transplantation. So, and these are animal studies where we have conducted, so there is another important topic, emerging topic is organoids. So now with the advanced stem cell research, we are able to culture, develop miniature small size retina inside our culture dishes. So those are called retinal organoids. And these retinal organoids are now a good source for transplantation. So before initially when we transplant, do the transplantation, so there were more ethical issues because the people tried to rely on aborted fetus eyes for harvesting the retina. But now due to ethical issues, which is not very encouraged, so now we are developing these kind of organoids in the cell culture dishes where you can develop small miniature eye structures that can be used for transplant. So this is a kind of a RPE transplantation approach where the retinal pigment epithelial cells are grown on artificial substrates and then transplanted into the eye. So these are some animal studies that shows the functionality of such transplants. And this is a kind of a retinal pigment epithelium transplant used for human clinical trial. This is one of the studies we have conducted in the clinic where age-related macular degeneration patients received retinal transplants. And this is again the fundus picture. It shows where the transplant is placed after the surgery. And we can monitor these transplants to confirm that they are surviving. So one particular study is already, the first phase 1 clinical trial is already over, and it's already started showing encouraging results. So this is about the retinal pigment epithelium, but when there are severe conditions when both the photoreceptors and retinal pigment epithelium is dysfunctional, in those cases, we have to transplant both. So for that, we recently developed a co-graft approach where it's kind of a tissue engineering approach where the retina organoids are grown in a cell culture dishes. Retinal pigment epithelium are grown on a substrate. We use a certain bioglues to attach them together and make it into a co-graft and used for transplantation. So the first proof of concept studies are highly encouraging, and we were able to show that we can make these kind of co-graft for transplantation studies. So the conclusion of this co-graft approach is it is possible to develop retinal pigment epithelium together with the retinal organoid co-graft for a transplantation. So we have established an implantation tool and a technique for making this approach that can be a promising technique to repair permanently damaged retina. When I say permanently damaged retina, means that retina in which photoreceptors are damaged, our retinal pigment epithelium is dysfunctional. So in that condition, one of the cell replacement approach is to make it into a co-graft and transplant it. So, okay, then coming to the next is vision restoration using retinal prosthesis. So on the left side, it shows about a patient who has a prosthetic device implanted into the eye. What you saw outside is the electronic device part. So I'll go to the detail in the next picture. So here you can see, so on the bottom, you can see that picture A is just showing the structure of the eye and optic nerve and its connection to the brain. On B, it shows how a small structure of the retina showing how different prostheses are placed. I already mentioned it in the previous slide. It can be epiretinal if it is outside the retina. It can be suprachoroid if it is outside the retinal pigment epithelium, or it can be subretinal if it is inside the retina. So the clinical trial we have conducted is based on an epiretinal prosthetic device. So the artificial retina or retina chip, the basic concept is a multi-electrode array is used to transmit the information obtained from a camera that capture the external images. Basically, a camera outside, it will capture the images. There is a device that converts these images into electrical signals, and using a cable, it goes into a multi-electrode array placed inside the retina. So this multi-electrode array stimulate the neurons. Since the photoreceptors are gone, the multi-electrode array can electrically stimulate the neurons. By electrical stimulation, it can produce neural signals that will comparable to the photoreceptor stimulation. And those signals can be taken to the brain to get a visual perception. So the resulting neural stimulation can provide a visual perception with a pixel size based on the number of electrodes used. So for example, if you use 1,000 electrode, multi-electrode array successfully and stimulate the retina, a person would be able to see another person standing in front of him. At least he knows what it is. But the maximum the current prosthesis is able to accomplish is only 60 electrodes. Because of the small size and complexity of the electrode array to be placed inside the retina, it's not possible to make 1,000 electrode. So, okay, basically a very low pixel size MEA is now being used. So when we do these clinical trials, we have to conduct preclinical studies in which we will try to stimulate the retina of a rat, especially normal rat and degenerate retina. And we will try to get the visual activity from the brain, the visual center of the brain, that is supracolliculus. So that is an electrophysiological recording. So that will, so you can put a, for example, I have done those experiments. You can put a small electrode into the eye of a rat, and you can keep it close to the retina and electrically stimulate it. And if you place an electrode on this brain, you can get that response. So that's how you confirm that the retina can be stimulated electrically. And that's also used for determining the stimulus specifications like amplitude or frequency of stimulation. All those things are first standardized to determine the safety and everything. Then only it moves to the next level. Usually how it works for a clinical trial is start with the small rodents, especially rats and mice. Then it goes to the next level would be dog or pig for a prosthesis, which is the real size for clinical trial. And just to see how the implantation and surgical interventions are going. Then it will go to the human. So this is just showing how the electrophysiological recordings are conducted. So usually when you stimulate the right eye, the recordings are made from the left side of the brain. That's how the optic nerve always crosses each other and go to the opposite poles. That's how the one eye is stimulated and the other eye is, the other side of the brain is used for recording the responses. Just showing the setup in the lab and different type of stimulation. And this is how when you record from the neurons in the brain, this is how the activity look like. So for example, here light response looks like. And then when we use electrically stimulate, you can see it's pretty much similar, like how it looks like a light response. That's how the electrical stimulation also looks like. So that shows that, as I mentioned before, when you place a multi-electrode array close to the neurons of the retina and stimulate it, you can produce neural activities comparable to a light stimulation. So this is the Argus 2 epiretinal prosthesis, which is now currently used for human, and now it is commercially available. So on the right side, the second side medical products, they produce this. This is a multi-electrode array I mentioned before. It is based on the surface of the retina, and just showing how a camera there capture, the receiver is the one which converts the camera images into electrical signals, and the retinal implant is directly stimulating the retina. The person can see the letters That's the concept of the epiretinal prosthesis. So if a person, more severe condition where optic nerve itself is damaged, you have to directly stimulate the visual cortex of the brain. That is called cortical visual prosthesis. So again, the Second Sight International Company, they developed a system called Orient system that is using the deep brain stimulation. So instead of having a camera stimulating the eye, so the Orient system will directly stimulate the visual center of the brain, and that can also help patient to get some kind of visual perception. Okay, so the another visual processes concept is based on ultrasound stimulation. So when you hear about ultrasound, everyone might be familiar about using ultrasound for finding the baby in the uterus. So that's what the ultrasound uses. But now the ultrasound is emerging as a new technology for neural stimulation. So we have a concept of developing a visual processes based on ultrasound stimulation. So because we already conducted single cell recording from retinal ganglion cell showing its activity during ultrasound stimulation. So we have conducted several brain electrophysiology studies showing that the retina can be stimulated with ultrasound. And just like electrical stimulation or light response, we can get responses from the brain in rats by ultrasound stimulation. So it's just showing the different types of parameters we used for ultrasound stimulation. And we also conducted experiments that ruled out that there is no light evoked activity. So the ultrasound directly activate the neurons, and it can help to produce visual perception in the brain. Then the next type of visual processes is cellular visual processes. That's another concept. So I already mentioned about stem cell therapy, which is a complete replacement of the damaged cells. Retinal prosthesis based on a multi-electrode array, that is a camera captures the images and directly stimulate the retinal neurons. Then ultrasound, instead of electrical stimulation, ultrasound waves can be used for stimulating the retina to develop a prosthesis. The advantage of ultrasound stimulation is it is less invasive. There is no need for any device to be implanted inside the eye, and it is more safe, but it is not so far developed up to the level of the epiretinal retinal prosthesis because it's in a very early stage. Actually, in the last 2 years, that's when it started to develop into a new technology, using ultrasound for stimulating the retina and restore vision in blind patients. Okay, then all those is over, and now I'm talking about cellular visual processes. So all this, the previous ones were about activating the retina as a whole. Now we are looking at individual cells inside the eye that can be developed into a prosthesis. So the idea is to engineer cells to convert. So I mentioned in the beginning that you can target specific cells in the retina, make them light sensitive. So retina, that's the concept. So there are different types of cellular processes. So for example, this one, light activated channels or transporters, for example, channel rhodopsin. And also just to think about, one example is just to think about the photosystem that is in the plant, which absorb light and do chemical changes inside the chlorophyll associated green plants. They can make all this conversion of light energy into whatever chemicals. So those kind of, if we can separate that light sensitive particles from the plants and introduce it into the eye, that can be used as a molecules for converting the light sensitivity into visual perception, especially in the case of when photoreceptors are lost. That is a concept. And there are other kind of approaches too. We worked on photovoltaic nano switches that will help to avoid foreign gene expression or gene therapy, and it helps to use different types of spectra. For example, when you use a photosystem 1, it has limitations. It's not sensitive to photosystem 1 is the plant-derived one. I have tried it actually, but we were able to get some results, but it has a lot of limitations, like it cannot be, it need a very strong light. Its half-life is very short, and also we found that the potential is very low. So we are moving to different things. So this is, the concept of using a cellular prosthesis. So the photovoltaic nano switches, or PVN, it's a chemical compound that can be introduced into the eye. You can inject it into the eye. It can bind into the neurons, and especially it can influence the voltage gated channels and cause a polarization, changes in the polarization, just like how a neural signal is developed. And that can be used for causing visual perception, neural activity, and then brain visual centers can be activated using photovoltaic nanoswitches. These photovoltaic nanoswitches, or PVNs, are sensitive to light. So when the light falls on them, they make changes in the retinal neurons, causing changes in the ion channels and depolarization, and changes in the polarization that cause neural signals. RubyPi is this is actually what we used. RubyPi, that is a chemical we used for our studies, and we found that it has the ability to make these kind of changes in the cause neural signals to happen. So we found that that has great potential for acting as a cellular prosthesis. And these are just experiments showing in the cells how they activate the cells and sense neural signals. So the same way we did electrophysiological recording from the brain after we injecting this kind of RubyPi into the eye at certain, like 1 day or 2 days after injection, and we found that it will produce signals. So the way we confirm it is we do the injections in blind animals mostly. There are animal models that mimic human disease conditions. So just like age-related macular degeneration or retinitis pigmentosa, these animals become blind, and once they are blind, we use these animals for conducting these kind of studies. So we found that when we inject RubyPi, it shows responses, but the responses are slightly delayed, which is not very encouraging. And also when you look at the, this is a normal rat with the amplitude of the response. That's how the firing is. We don't get it like even half the size of the light response. So that is a disadvantage currently we are facing. And also we found that there are pupillary light reflex. For example, if a blind person, the pupil, I mentioned that it is like a camera shutter. When there is more bright light, the camera shutter closes just like that pupil will close. But if the person is completely blind, there is no activity. The pupil closing and opening will not happen. Looking at the pupillary reflex is a good indication of whether there is light perception or not. That's how we use this is for animal studies. For example, a blind animal, when we try the pupillary light reflex, like showing light in front of the eye and check the pupil size, it won't change. But we found that when we used the RubyPi, which is a cellular prosthesis, injected into the eye, it started pupillary reflex. That shows that, indicates that the signals are going to the higher visual centers of the brain that initiate the this kind of light evoked activities. So this is the last slide, and I would like to thanks to various collaborators working with me. Dr. Mark Humayun is a world famous ophthalmologist and a scientist. And Dr. Magdalene Cellular, she was once my mentor and a long-term collaborator. And others at USC and other institutes, we all work together for various research projects. Binlin and Deepthi are postdocs working with me. And the research support from a lot of different funding sources from US. Thank you very much. And if you have any questions, you can ask.
Thank you, Dr. Thomas, for the insightful and wonderful talk on the advanced technologies for vision restoration. You have covered all the advanced perspectives, which include the electrical stimulation studies using rat disease models and impact of ultrasound stimulation and the cellular visual prosthetics and their concepts and their goals and many more other applications in this vision restoration aspects. Now, dear participants, we are having the question answer session. If you are having any questions, you can put it as a chat box or directly you can unmute and you can ask. Meanwhile, we will be sharing the feedback link also, and please note, you have to fill the feedback link very carefully and to get the certificate for this session. And the certificate, like the conventional webinars, we will not be releasing the certificate soon, and it will be the participation will be thoroughly validated and will be releasing the certificate. If you are having any question, you can put it in the chat box or you can ask directly.
Hello, Dr. Biju.
Yes.
I am Dr. Sarla Gopalakrishnan from the Department of Microbiology. It was a very interesting and informative session. I have one question. That is, I experienced a macular degeneration, which was actually not age related because I experienced it at the age of 33. Okay, and I consulted with the ophthalmologist, and they identified it as macular degeneration and had a laser surgery.
Okay.
And at that time, I think there was no such, I hadn't any information about such newer technologies. But they said because of this laser surgery and how these rest of these cells can be saved and undergone that. So if I didn't do that laser surgery, what now happened? Whether it is progressing to complete blindness or what is your?
So let me say, basically I am not a clinician, so and that's one thing. So I am not in a position to give a right answer for you, most probably. The second thing is I don't have any, even though I am not a clinician, since I am working in this field for such a long time, I have seen patient data. Sometimes people approach me asking questions about the same way you asked me the question. So I asked them to send me the clinical report and I evaluate it, and that gave me a better insight into the disease condition. But here, both these conditions are not available. So based on what you told me, my understanding is you might, the laser treatment was mostly for some kind of blood leakage, probably. There could be some blood leakage and they try to stop it by using the laser. Otherwise, I don't see any other use for laser for age-related macular degeneration. So it is called wet AMD, where there could be blood vessels growing and they might have used laser to stop it. So I think if that's the case, I think that's a good intervention for at least slowing down the progression of the disease. But unfortunately, most of the eye diseases, unlike many other diseases that affect human being, even the case of cancer, we know that like even the blood cancer, there are certain, if the treatment is in the right way for the right person, it will completely, cured. But eye diseases, it's always a challenge. So unfortunately, once you get some kind of diseases, there is no way you can completely cure it. But because of the modern medicine, there are several approaches that help to slow down the progression of the disease. That's what most of this, the treatments are meant for. And on the top of that, also, I mean, I just wanted to, so although I explained about different disease, I mean, treatment strategies, like I already told that there are 2 of them already in the clinical trial. Still, all these are in a very early stage. Eye is a very highly complex organ in the human body. It's not easy to fix the issues. It's very complex. So we are still in a very early stage of curing blindness. So these are different approaches, but at this point, I can tell you that there is no treatment in the world to restore vision after it is completely gone. So the laboratories like my lab and the several labs all over the world, they are trying different approaches, and they are also showing some proof of concept, but none of these are 100% effective for restoring vision.
Thank you very much. And one more question. That is this age-related macular degeneration. Generally, on an average, it starts in which age?
So, sorry, so your question is what age it starts? I never heard about it starting so early. Usually it is above the age of 60. That's what age-related macular degeneration starts. And again, it's highly hereditary, and also it depends on a lot of different factors are involved in it. For example, environmental pollution. If you smoke tobacco, your diet is, if it is high cholesterol diet, if your living habit is unhealthy, like you don't no exercise, eating a lot of oil fried things, all these things can contribute to the disease. But generally, it's above the age of 60.
Thank you, sir.
Any other questions from the participants, especially students?
Hi, sir. I'm Anupama, a student from Life Science. Sir, you have explained us various technologies for vision restoration, and I would like to ask you that, can these technologies be used as a treatment method for blind people to restore vision?
Yes. For example, the retinal prosthesis, that's already, if you recollect, there was Argus 2 implant. So that one, we have already implanted it in completely blind people. And we found that, so when you talk about completely blind people, we have to think about their situation. They don't even know when the daylight is, or when the lights are turned on in the room. So there is no difference between darkness and lighted environment. So illumination and darkness, they cannot even discriminate. That's a very terrible and disappointing situation. So any kind of approach, even up to the level of showing a light perception, is a big achievement. So the visual restoration based on retinal prosthesis was able to make a person to read very large letters, and also to see there are limitations, like a person probably by training, he would be able to see a white cup sitting in a black background. So this kind of like high contrast and big size things they can see. That's the maximum we were able to accomplish. And as I mentioned before, the pixel, the multi-electrode array, number of electrodes, those are the limiting factors. So at this point, that's all the accomplishment for vision restoration is retinal prosthesis. Other than that, so far I know there is no other strategies even went to a clinical trial to prove that a blind person can see something. Is it clear?
Yes, sir. Thank you.
Any
Good
other
morning, sir.
Mm-hmm.
Can we use these technologies for treating diabetes retinopathy?
I would say depends on what stage it is. If it is a severe condition where in diabetic retinopathy after that's what we call it irreversible blindness. That means all the neurons are completely damaged and there is no way you can use any kind of treatment strategies to rescue it. In that condition, irreversible blindness occurred, and that condition, the only scope is, at this point, retinal prosthesis. But again, I forgot to mention it, retinal prosthesis is, even though it is commercially available, I think it is around 2 lakhs US dollars for at least 1 to 2 lakhs US dollars you have to spend for getting one of these retinal prostheses. And even with that, what you are gaining would be very limited level of vision restoration. So any kind of advanced disease condition where the blindness is complete, I would say that whether it is diabetic retinopathy or age-related macular degeneration or retinitis pigmentosa, there is not much scope now except for some minimal vision restoration with this highly expensive retinal prosthesis.
Hi, sir. I'm
Yes.
Gidu. Sir, is the procedure is painful?
The procedures, especially in the eye, it will be done in the anesthesia, and I don't think it is very painful, but using it is a pain actually. So the device, although it looks very simple, it's very complicated, and it's not like you can get it implanted today and tomorrow onwards you can start using it, walk around and see things. It's not like that. It has a lot of limitations, like you need to get the training and you may not be turning it on always. You might have to take out everything, and majority in 24 hours you might be using it for only a few hours. It cannot be used like throughout the day and night. So that's a more pain, not the pain due to the implantation of surgery.
Thank you, sir.
Hello, Dr. Thomas. I too have a question.
Yes.
You have already shown about some data about the image analysis. Generally, in the this type of visual damages, there are a lot of images people used to analyze by using advanced computational biology tools. As our program is life science and computational biology, I think students will be interested to learn whether you are doing the image analytic aspects and what are the computational biology tools you are using to analyze such a kind of images for the diagnosis of this particular process.
That is a highly already emerged and established in the US, the artificial intelligence kind of thing for image reading. And many of these eye research centers, they have their own image reading centers. I'm not involved in that, so I don't know what kind of software they use, but I know that they all use the fundus image. Even that is helpful for identifying the disease condition, and so that is widely used and highly established here. But I'm not sure what kind of tools they use.
Okay, thank you very much. Is there any other questions from the participants?
Good morning, sir. I am Akshaya. What is its role in the treatment of color blindness?
Can you repeat the question again?
Yes, sir. What is its role in the treatment of color blindness?
The treatment of color blindness. So I didn't get your question again. So what kind of treatment? That's your question. Is that the question? What kind of treatment available for color blindness?
Role in the treatment of color blindness.
What kind of
role?
Role? Oh.
In the treatment of color blindness.
For color blindness, unfortunately, I don't think there is a effective treatment, because it's when you look at a blind person or a person who is facing blindness, the color blindness is not considered to be that serious, I think there could be a way of negotiating with that defect by other ways, because it's not considered as such a serious condition actually. So I'm not sure what kind of, yeah.
Thank you, sir.
Okay.
Good
If there are no more questions. Teacher, കേൾക്കുന്നില്ല. ടീച്ചർ പറയുന്നത് കേൾക്കുന്നില്ല. ഷിജു ടീച്ചർ.
Is it available in India?
Which one? Which one?
This methods can be used here.
Oh, which one? I didn't hear you. I don't think any of the clinical trials, what we are doing here is available in India at this point. But I know that stem cell therapy, several laboratories in India, I am also helping in PGI, Chandigarh. So they have an investigator who is working on it. And also there are a couple of other institutes in South India also trying to develop stem cell approaches for treating eye diseases. But I don't think any of those moved to clinical trial yet.
Good morning, sir.
Okay,
I'm Jyotish
please,
Matthew.
please go ahead. Yeah.
How it is useful in treating night blindness?
Night blindness is, as I mean, color blindness, night blindness, these kind of night conditions are less severe compared to severe blindness. For example, any of these treatment strategies like prosthesis or stem cells or the other things I mentioned, these are not meant for someone who can negotiate with whatever vision left for them. These are all meant for people who are really blind, because nobody wants to interfere in an eye which has maybe 50%, even 20% functionality. Nobody will like to enter into any of these treatment strategies, because there is no guarantee that eye is very sensitive. You might even end up with losing whatever you had before. So the research is mostly aimed at vision improvement, not improvement, vision restoration for completely blind person who don't have any other scope in their life to see light. So that is what most of this research is now focused on, because when a person is having 10% normal vision left, none of these strategies would be, may not be beneficial. It could be even harmful. They might end up with even losing that. So I don't think these kind of color blindness or night blindness or those kind of diseases are considered to be that severe to try any of these clinical trials, what I explained today.
Thank you, sir.
Good morning, sir. Does it have any side effect?
All this will have, you know, eye is very sensitive. So if you try to put anything inside the eye, there would be a lot of reactions. Any kind of intervention, stem cells, gene therapy, retinal prosthesis, of course, all this will have side effects. Side effect in the sense not going to affect your health condition, but it can cause some problems eventually inside the eye where you injected it or you placed it.
Okay, sir. Thank you.
Is there any more questions? If there is no questions, then we can go ahead to the next session. So thank you, Dr. Biju B Thomas. Thank you very much. And I over to Vaishnavi Shivadas. Vaishnavi.
Yes, sir. Thank you all for your wonderful cooperation. Now I request Dr. Sarala Gopalakrishnan, Assistant Professor and Head of the Department of Microbiology, for the vote of thanks.
Good morning, all. Am I audible?
Yes.
So we were listening to a very informative and useful session by Dr. Biju B Thomas. Now, on behalf of the Life Science Department, St. Pius College, and all the participants gathered here, I would like to express a very hearty thanks to our resource person, Dr. Biju B Thomas. Actually, he started from the structure of the eye and have gone through all these important aspects of the current trends and developments in the technologies in the visual restoration, including these retinal organoids, co-grafting, then visual prosthesis. So all these things, and even he mentioned about the ethical issues regarding using this fetal eye during the experiments. So thank you very much, sir. Thank you for coming over here and presenting this talk before our audience. Now, I would like to thank our principal in charge, Dr. Sri Biju Joseph, sir, for arranging such a session, and also for the organizing committee for bringing such a resource person here. Thank you very much. Now I would like to thank Dr. Shiju Jacob, Head of the Department of Life Science and Computational Biology, for arranging this session for life science students, and also for these microbiology students and UG and PG students from other departments and colleges, our alumni, etc. And last but not least, I would like to thank all the participants gathered here, these presenting students, all the students, the faculty members, and all the participants. Thank you very much. Thank you, one and all.
Thank you, Biju.
Thank you, ma'am. Once again, thank you all for your wholehearted participation. The same participation will be expected in the upcoming sessions as well. Thank you.